I introduce our works on ThreaDom. Our aim is extracting the domain boundaries from multiple threading alignments. Okay. Protein modular is composed of domains. Assigned domain is important to efficient elucidation of protein functions and evolutionary mechanisms, even if not absolute. Currently, the experimental determination of protein structure is often time and manpower expensive, and some proteins are not resolved today. Then the computation methods is highly demanded today. Okay. There are three categories of computation methods to prediction domain boundaries. The first category, we call it statistics or machine learning methods or ab initio methods. This kind of method implicitly be applied to any proteins, but all over the accuracy is low compared with homology-based method. The homology-based method is have higher accuracies when the closed template are identified, but the accuracy decreases sharply when the sequence identity is low. Especially, for example, the identity is less than 30%. Some prediction based on homology-based method will fall. The third category is I call it 3D model-based method. This kind of method need time-consumed simulation, and sometimes this method decreased with the size of protein target because the limited ability of the ability method foldings. Okay. We developed the ThreaDom. We aim to predicting the domain boundaries to the query sequence. There are no templates deposited in the PDB libraries. We want to give the researcher automatic pipelines we can use to determine the domain boundaries automatically. The third, we apply the online server and the source code is available for the academic user. Then, to this method, there are several key problems. The first one is the multiple threading alignment is based on Lumens. Lumens is a meta server which collects templates from several threading algorithms. The second is we need a map adapter for the different threading algorithms shown in this picture. You can show here. There are several threading algorithms. They use the different structure library. These libraries are different, especially in residue numbers. When aligned the template to the query sequence, the different regular numbers will give many difficult. Then the map adapters can help us solve this problem. The third is we designed a domain conservation score. Using this score, we can design the domain boundaries. The final step is detecting the discontinuous domains. In fact, there are about 10% protein sequence has one or two or more discontinuous domains. Okay. Here is the limits. The limits is developed by John Lambs, and the collected template like this. This is query sequence. Okay, typed 1, 2, and typed template M. This template come from multiple threading algorithms. There are three kinds of information can be used to determine the domain boundaries. The first kind is domain information or domain definition or the template, which is given by, for example, conf, scope, and others. The second is the internal gap. The template, there are some regions are missing or insert to the query sequence. The third is the terminal gap. The template maybe is shorter than the query sequence. These three information can be used to combine a domain conserver score. Like this, the domain conserver score is here. The two items is describe the template penalty score and the gap score. The gap penalty score includes the internal gap score and the terminal gap score. This is a curl domain conserver score. Okay. For this region, the only means this region, this residues may be not conserved. Give a cutoff here. There are 1, 2, 3 putative linkers. In this research, the middle of the linkers is used to cut the sequence to several fragments. If we suppose a domain is composed of one fragment, then this is final result. But in some cases, there are several fragments is merged into one domain. Then the next step, we should to detect the discontinuous domains. This is strategy for detecting discontinuous domain. The first step is use the domain conserver score to predict the boundaries. The second, we should detecting whether this query sequence is contained a discontinuous domain or not. The third is clustering the discontinuous domain template. There are maybe many, more than 15 templates. We cluster them to different clusters. The first cluster is chosen to refinement or substitution the domain prediction in the first step. Here is an example. This sequence is submitted by user to the web server or the ThreaDom. Okay, this is the predict result. This is the conserver score. If we give a cutoff, we have 1, 2, 3 cut position. Then we directly determine the prediction and the fault domain sequence. Using the detection discontinuous domain function, okay, we can combine or merge these 4 segments into 2 domains. Here, we must mention that a new small fragment is added. I must say that for ThreaDom, we designed them. We can't prediction the boundaries which is close to the terminal, the distance within 40 residues. Here is very short fragment, but use the cluster or detection the domains, we can get that. That is a measure. We test or benchmark this using training data set and test set. We train some parameters, mainly in this part, there are several parameters needed trained and test on the other data set. We choose the 1, 2, 3, 4, 5 controlled measures. The first 2 is a homology-based measure. These 3 is called ab initio measures. We categorize the test targets into 2 categories. Also, sorry. Oh, no problem. One is for easy targets, another is medium or hard. For easy targets, that means use threading algorithm. There are more than we choose more than each template is a good template. What's good template? Good template means the law means think. There are more than we choose 8 threading algorithm. If there are 8 threading template, more than 8, then we think it is a good template. Else, it's not. The first test is a single domain or multiple domain classification. We can find that for the easy target or medium hard target, ThreaDom do the best. Here I want to say that for easy target, ThreaDom then better than the 2 simulacra-based measure. For the hard or medium targets, ThreaDom is better than the other 3 ab initio measures. For the boundary informations, here is the result. For the 3 categories, ThreaDom does ranked first. 1 for NDO score, DBD score, and the precision. In the record of the boundaries, here for hard and medium prediction, ThreaDom is a little lower than the PPR. Okay, this is result on the test data set. In the domain control score, we have 3 items. That is template, internal gap, and terminal gap. We test this item, this component, and we found that we use template and gap together. We get a better result. Here we show examples. This target is a hard target. Is its domain definition is and residue 55. Our prediction position is 30, 60. Okay. From the aligned template, we can find the picture BCD is the distribution of the terminal gap, internal gap, and temporal domains. If we even depend on these three pictures, we can't tell which position should be cut the sequencing to several domains. But when combine them together to the DC scores here, we use the cutoff 0.8. We can cut the sequence into two parts. Then we get the position 60. It's a little errors to the definition. This is the case for discontinuous domain predictions. I must say that the DCD scores, the domain control score, then cut the sequence into different fragments. If there are several fragments into one domain, we need a method to detect. In this target, the yellow and the red is in one domain, the green in another domain. In this part, there are two boundaries. Okay. The prediction is cut the sequence into three fragments. Then we classify the target and the discontinuous domain first. The next step is cluster the template. The first cluster is used to replace the boundaries practice and the first step. Then the final result is like this. This is another study, a case of CASP 8. A and B is a free model target. P1 and P2 is the definition of the target. The predicted result is here and here. There are little residues errors. For this is a discontinuous domain proteins. In fact, there are ThreaDom cut this sequence into 1, 2, 3, 4, 5 fragments. Use the domain detection. We correctly successful detect that there are two domains, the green and the yellow, the green and blue in domains, and others in other domains. Okay. This is another test compared with the measures in CASP 8. In our research, in our benchmark, we found that for free modeling, multiple domain hard, discontinuous domains, ThreaDom do good. Okay, there is a problem is that for the free modeling test targets, ThreaDom is a little lower than Zhidong. Okay. The average and the discontinuous domains and multi-domains is ranked first. We also test the measure on the CASP 8, the dataset of CASP 8 and 10. Here, we given two strategies for ThreaDom. The ThreaDom 1 and ThreaDom 2. ThreaDom 1 is predicted the boundaries directly uses the domain conserver score, not without the discontinuous domain detection. ThreaDom 2 with the discontinuous domain detection. We found that for the between categories, contain targets, ThreaDom 1, 2 ranks the top 2 here. In this, it's much better than other methods. In case 9, we found that there are similar results in multiple domain categories, but in free modeling and hard concept 9, the recall and the DBD scores is not the best. In total, I think ThreaDom have a reasonable performance for this test. Okay. I think we also found that there are some drawbacks of our measures. Here, we give some domain conserver score. The different cutoff will give why the cutoff gives the wrong prediction. The cutoff is trained use the trained data set. That's a global cutoff. It can't deal with all the situation. In this picture or in figure A, if the cutoff is here, there is only, then we decide this sequence and two domains. In fact, this target is a hard target. There are no good template. Most templates is terminal here, or some template have a boundary here. Then the final result is wrong. In figure B, there is one only two peaks. When the cutoff is more than this peak, we think the ThreaDom algorithm think there are only one domains. In fact, for these targets, there are two. The main reason is that from 0 to 45, okay, this part is very short. In ThreaDom, we will penalize residues around the gap of the boundaries of template. These peaks is weakened sharply. But if we use template a lower cutoff, we will get the right boundaries. For these pictures, see, this is a long sequence. For threading alarm measures, some template is shorter than the query sequence. This gives a distribution. We found that there are no templates more than 300 residues. For this part, that means no enough template information for determining the boundaries. Okay. Some errors may be deduced by the template inconsistency definition. For here is example. This is a query sequence. This is the homologous template. They have different domain definition. The final result is wrong. Okay. ThreaDom, you can use it online to download the source code. This source code depends on Artisan 2.1. For the long sequence, for the user, I selected that. If a user found this domain conservative score, you can cut this part and submit it again. Then the final result may be like this. Finally, combine these two results together for the whole sequence. Okay. Then we think ThreaDom can do better than ab initio measures and machine learning measures without closed homologies. And it not require the lengthy modeling simulations and not limited on the size of protein targets. ThreaDom can detect discontinuous domains automatically. I encourage the user to use these measures. Thanks the authors Dong, Yan, and Yang. Thanks the fundings and the reviewer and the others who help us. Thanks for your attention.
Okay, we have time for a question or two. Okay, so I'll start. So, what? No, I have a question.
No.
So, what do you think you can do to improve the method further? How can you improve your method further? Do you want to speak into that?
Okay. The method is developed aimed to. There is no closed homologous. In use our methods, we can do. We can solve. For example, if you use C++, can't find the homologous template. Use our method, I think it will solve your problem well. I think.
Okay.
Okay. Thanks.
Okay, we have one more question.
Hi. Have you thought of also identifying the critical residues in the domains?
Singular?
The critical residue for the interaction with their targets?
No. Can you?
What I mean is that you also want to work later in the direction where people can really find out which residues of the domains are really important for function.
Yeah. For the we have planned to develop the methods. In fact, we want to do more on the discrete domains. Have no plan on the develop some measures of the